Welcome back to Biochem 2, Chapter 8. So this is going to be a little bit different of a chapter. There's a decent amount of information here that's just kind of in graphs and charts and diagrams. So a lot of this is going to kind of be working it out on your own, figuring out exactly what there is and like the names of everything and how they're connected. So this is the biomolecules for one carbon group transfer reactions. Tetrahydrofolate, vitamin B12. B12 is our cobalamin. Tetrahydrofolate is related to B9. And S-adenosyl methionine, also known as SAM, S-A-M. So at the completion of this lecture topic, we should be able to, 1, explain the importance of the following coenzymes, particularly the specific biochemical reactions in which they participate. So we have tetrahydrofolate, aka THF, aka vitamin B9, vitamin B12, and the S-adenosyl methionine, SAM or SAM. We want to know the sources of THF, vitamin B12, and SAM, where we get those from. Describe the general reactions and functions in which these coenzymes participate. Remember, coenzymes, different than our cofactors. The cofactors are the different types of metals. Coenzymes are vitamins. Demonstrate the relationship among THF, vitamin B12, and SAM. So, many biochemical reactions involve the transfer of a single carbon group from one compound to another compound. These carbon units can be present in different oxidation states. This is going to be the key kind of word that we're going to be looking at, oxidation states. A lot of our questions and things are going to come back to kind of what's the oxidized or reduced form of these different vitamins and what state are they currently in when they do different things. That's kind of what we're going to look at and where you're going to want to focus some of your studies. So the most oxidized form, CO2. Okay, specifically these are those one carbon groups. The most oxidized form of single carbon groups is CO2, and that is transferred by biotin. Remember, biotin is also one of our B vitamins, and that is going to be our B7. We have one carbon groups, such as C1, at lower oxidation states than CO2, and they have different carriers. Carriers meaning those coenzymes. So the key carriers of one carbon units that participate in a lot of these different reactions, which involve transfer or addition of the one carbon units, are these three: the tetrahydrofolate, the vitamin B12, and the SAM. Starting with tetrahydrofolate. So its major function, and this is where we really want to lock in and focus on. The major function is to transfer single carbon group or one carbon units, which are present at various stages of oxidation and attached to the tetrahydrofolate. And remember, tetrahydrofolate is B9. So this is produced from vitamin folate or folic acid. So THF is the active form. So folate or folic acid is the inactive. So THF is the active form of vitamin B9. It is a water-soluble B complex vitamin. So this is where we get into this whole kind of big chart, which kind of shows us the pathway of all of this, okay? So we have tetrahydrofolate being kind of our active form of all of this. And we have a bunch of enzymes and all this stuff going all over the place. I'm not going to spend a lot of time in this video going over this at all because I think it's going to be better when I do one of the live sessions, I'll do the Linus learning questions, and that will really hit home what you need to know from all of this rather than trying to go through all of it right now. So make sure you look out for that next video, going over those Linus learning questions. So folate cannot be synthesized by humans. If anybody knows somebody who's been pregnant, they know folic acid, B9 vitamins are super big with pregnancy because we cannot make them ourselves. Our body cannot make this vitamin itself. So we need it when we're growing a new human because it's essential. It's very, very essential for our nervous system development. If you don't have folate in diet, that's when children are born with deficiencies and issues as far as like spina bifida, where their spinal column doesn't properly form. Okay, so it's essential vitamin, essential meaning that we have to consume it, and it is obtained from the diet or the intestinal microbes can synthesize it. Folate is a doubly oxidized form of THF. So that means, so we have THF, oxidize it once, oxidize it twice, and then you get folate. Okay? And in that doubly oxidized form, folate, it cannot transfer those 1 carbon units. It has to be reduced twice to generate the active coenzyme form, which is the THF. That's what we want to remember. Inactive form is the folate or folic acid. The active form is the THF, the tetrahydrofluorate. Okay? Tetrahydrofolate. Active form. Okay? And that, we'll see, he might ask a couple questions about how many stages of oxidation or reduction need to happen for that. Okay. Again, we'll come back to these kind of graphs and charts with the Linus learning questions as they come up. So the reduction of folate forms the active coenzyme, THF. The reaction is catalyzed by the enzyme. This is where we're going to want to remember dihydrofolate reductase, and the reducing agent that is used is our NADPH. And remember, our reducing agent is the NADPH, because we're going to be when we're going from folate to tetrahydrofolate, have these double arrows, we're going this way, and so we're giving up and giving those hydrogens to get the tetrahydrofolate. So the first reaction, the folate is converted to produce dihydrofolate by dihydrofolate reductase, which requires an NADPH. The second, again, uses the same enzyme, so make sure we know that enzyme, to generate then the tetrahydrofolate using the NADPH as the reducing agent. Folate is mostly present in our body in its reduced form THF. It doesn't make a ton of sense for it to just be floating around as folate. So with B vitamins as well, we can consume B vitamins in excess because they're water soluble. So if you take an excess of B vitamins, you're just going to pee them out at the end of the day. So if you look at most like multivitamins, a lot of supplements, they tend to have like 1,000% of B vitamins, when in reality it's not super beneficial because they just go right through you. Okay, so oxidation and reduction of the 1 carbon groups in THF. So now we're going to look a little bit closer at the actual tetrahydrofolate, kind of what's happening there. So the 1 carbon groups transferred to the tetrahydrofolate are linked covalently either to N5 or N10. So that's a nitrogen in the 5th position or a nitrogen in the 10th position of the tetrahydrofolate molecule. or they form a bridge between the N5 and N10. These guys right here, those positions are going to come back up. So make sure we write those down. Remember, hey, those N5s, N10 positions, just where those are located. N5 right here, N10 right there. We're going to focus on those when we talk about the 1 carbon groups that we're going to be transferring. Tetrahydrofolate obtains 1 carbon units from the following compounds. This is another place we're going to want to remember. This is where some test questions are going to come. We have serine, glycine, histidine. Remember histidine we talked about getting turned into histamine, formaldehyde, and formate. Here's all of those sources of the 1 carbon unit. We get tetrahydrofolate going in there. Then we have these different precursors, and then they make the products. So the 1 carbon groups can either be oxidized or reduced while they remain attached to the THF molecule. This is shown in this figure here, so either they can go either way, right? All of them are going to be interconvertible by the enzymatic redox reaction. The 1 carbon units may be at the oxidation levels of formate, formaldehyde, or methanol. Just remember our oxidation reduction. Oxidation is loss of electrons. Reduction is gain of electrons, the type of questions that we're going to do. So if we're gaining electrons, also gaining hydrogens. So our formate is oxidized, and the methanol is going to be reduced from formaldehyde compared to, so we look at the number of hydrogens, take away hydrogens, we're taking away electrons, being oxidized, adding more electrons, adding more hydrogens, we are reducing it. So here's where kind of this chart comes in, and we see that as well kind of in the PDF form. Dr. Sarkar puts a lot of these graphs and stuff already in here as well. So we're going to kind of see, so you can see the most reduced is the methanol, the most oxidized being the formate. So we'll see these. This is kind of the, we'll look and see again kind of what questions we have on the Linus Learning and how deep to go into that specific chart. So THF participates in the transfer of the 1 carbon units. Deoxyuridine monophosphate, DUMP, is going to be converted to deoxythymidine, or also known as DTMP, catalyzed by thymidylate synthase enzyme. This is going to be one of the enzymes, as well as the reactions, especially DTMP. We're going to want to remember those in the future. Okay, so we transfer that 1 carbon unit from that N5, N10 region on the tetrahydrofolate to the DUMP in order to form the DTMP. And so there we have our deoxyribose DUMP, deoxyribose DTMP, in that little process view there. Again, we'll focus more on this, kind of the ins and outs of this, with the Linus Learning questions. So these following 2 compounds primarily function as a little clinical correlation. The following 2 compounds primarily function by inhibiting purine biosynthesis. Remember that word purine. Purine and pyrimidines, those are our nucleic acids that we use to make DNA. So that purine biosynthesis, and therefore have the ability to slow down cancer cell proliferation. So they can inhibit the making of those purines of the DNA components, and because of that we can slow down the cancer cell proliferation. So methotrexate is a structural analog of folate, meaning it's similar in structure to folate, that inhibits the dihydrofolate reductase. 5 fluorouracil, a lot of these words. Fluorouracil is a blocker of thymidylate synthase. So want to connect those two, so the dihydrofolate reductase. I'd even come up here and say this thymidylate synthase enzyme, this guy right here, 5-uracil, inhibits this enzyme. And we'll keep going. I apologize for the pauses. My cat decided it was a great time to jump up onto my face right before I started recording this video, so got a little dander going on. Again, this whole chunk, this whole component, we'll kind of work through it as well. All of these are the ones that we talked about: the serine, the glycine, the histidine, and this is just how all of these get connected in the formation of purine biosynthesis. So see how all of this comes all the way through there in order to form the purines. And also how glycine, can be converted to serine. We can go different ways. Okay, so that's our tetrahydrofolate. We'll continue to look at that and see kind of what's going on there when we get to the questions. Vitamin B12. Vitamin B12, or cobalamin, it's not synthesized in higher animals such as humans, so the major source that we get from it is our dietary: meat, eggs, dairy, fish, poultry, seafood. All those animal structures have the vitamin B12 in them. So the structure of vitamin B12 contains a corin group, which is similar to a porphyrin ring, which is found in heme. We talked about hemoglobin a decent amount last trimester when we talked about kind of the structure, primary, secondary, tertiary, quaternary structure, that kind of stuff. Difference between heme and vitamin B12 is the cobalt at the center. Cobalt is at the center instead of iron, which is in heme. So the functions of vitamin B12: transfer of a CH3. Remember our functional groups, what the CH3 is. That's our methyl group. CH3 is our methyl group. So methylation, which we know has to do a lot with kind of, activation of stuff, or inhibition of stuff. So the transfer of this methyl group from N5-methyl tetrahydrofolate to homocysteine to form methionine. And I had mentioned this at one time. I don't recall which lecture, which chunk of material we were going through when we talked about this, the homocysteine and methionine. One of these earlier chapter, a little earlier chapter, we mentioned those. While I wait for that to open, we'll keep going. The homocysteine and methionine kind of has this loop that's very, very important when it comes to maintaining the body's homeostasis. Let's see. So homocysteine remethylation. We have food. The food produces methionine that gets demethylated to homocysteine, which can then go off into its own process, but from homocysteine, B9 transfers a methyl group to the B12, and it's going to remethylate it into methionine. So B9 gives methyl group to B12. B9 deficiency affects the B12. And so the B9-B12 relationship is super, super important, because this has a lot of effect on, and actually on our blood, and a couple of other factors where if we don't have enough B12 or B9, we can get different types of anemia, meaning that our red blood cells aren't functioning the way that they're supposed to. We don't have enough of them. Okay. Either don't have enough of them, or they're actually like the wrong size or not functioning properly. So that was one of its functions, is to work with B9 to form methionine, and also the rearrangement of the methyl group of L-methylmalonyl-CoA in order to form succinyl-CoA. And that's the main function of vitamin B12. Now our SAM, S-adenosylmethionine, it is a key methylating agent in several reactions in which a transfer of the methyl group from THF is not favored. So basically, SAM is our secondary guy, where if THF can't get it done, meaning that they're like, just because the reaction doesn't work out nicely, SAM is going to be the one who does that job. So it's going to add that to either add that methyl group to either oxygen or to nitrogen. So the synthesis of SAM and its breakdown, it is synthesized from the reaction of ATP and methionine. We see that adenosyl methionine. So as with activation of vitamin B12, ATP donates the adenosine. So we can look right here. Methionine, the ATP, methionine adenosyl transferase. We got some enzymes and stuff going on here. We get the S-adenosyl methyl 9 and different methyl transferases to give us a S-adenosyl homocysteine to homocysteine methyl tetrahydrofolate, and kind of go all throughout this little loop. Again, we'll come back to this loop and these kind of graphs if we have specific questions about them in the line of learning, because that's typically the best way to gauge which information is the most important. So with the transfer of the methyl group, SAM forms S-adenosyl homocysteine. That's adenosyl methionine. It forms the S-adenosyl homocysteine, which is subsequently hydrolyzed to form homocysteine and adenosine, and that's the relationship among these three is now shown here below. How these guys are all are interconnected. So here, FH4, this guy, this is going to be our B9. What's going on with the B9? The B12. And then this other reaction converting the methionine to the homocysteine, which then needs to get methylated there, around. That's how all of these are connected within this reaction. That demethylation of the methionine is coming from SAM. And so why is that important? Because so essentially we're essentially passing that single carbon group to each of these in order for them to do the job that they have to do. So if we need B12 cobalamin to do specific things, to go and interact with specific things and perform specific interactions, we give that one carbon unit. If we need folate for specific things, we leave it with folate, and folate goes into this thing. If we need it for something else, we get it through from the 9 to the 12 to the methionine, and then which then gives it to the SAM, and it gets it and goes to what and does what it needs to do. So, for example, all of these guys down here converting norepinephrine to epinephrine, really, really important one. You'll also probably see this one, the acetylserotonin to melatonin. We need SAM involved with all of these reactions in order for them to happen. Okay, so that's just a little overview of these 3 coenzymes. We're going to come back to this when we do the Linus. We should be doing that here shortly, and I'll have that video posted up as soon as we go through all of those questions and kind of dive into the specifics of each of these. Thank you guys for watching, and have a great day.